hizo el trabajo fin de máster, fin de grado también, no.
No, no había fin de grado en ese momento.
Fin de máster y la tesis en nuestro grupo, bajo la dirección de Vicente Mariscal, que está aquí, y mía. Y yo lo que destacaría de Félix, que fue una cosa que estuvo muy clara desde el principio, es que, bueno, aparte que es un tipo muy talentoso, que él declaraba que le encantaba la microbiología desde el minuto cero. Y de hecho, haciendo la tesis ya estuvo claro que su postdoc iba a ser en bacteriología, y a mitad de la tesis ya tenían localizado y acordado el sitio para hacer el postdoc, que lo aceptaron sin ningún problema, en el John Innes Center con Mark Wagner, para trabajar en postdoc. Me consta que ha estado trabajando en varios temas allí y nos va a presentar hoy lo que considere más oportuno para ilustrarnos de lo que hace allí. Félix, muchísimas gracias por haberte prestado a dar el seminario. Él le ofreció contarnos sobre el grupo que hacía y realizaremos un seminario al instituto.
Vale, gracias Enrique. Bueno, esto es como reunión de amigos, como dije antes. Y bueno, voy a dar el seminario en inglés porque me es más sencillo un poco hablar de los nombres de las proteínas en español. Creo que podrían ser un poco más complicados que en inglés. Y bueno, yo os voy a contar un poco lo que he estado haciendo estos casi 3 años ahora allí en el John Innes. Y bueno, pues voy a contaros. Y como podéis ver, bueno, a ver esto. Se supone que esto tiene un cursor, pero también trabajo con un bicho verde, ¿vale? Aunque no fotosintético. Okay, so I'm switching now to English. So I'm working with Streptomyces, and I'm sure that not everyone here has heard about Streptomyces because it's not very common, because I didn't hear about Streptomyces before. So Streptomyces are actinobacteria, so they are bacteria that they live in the soil, and they are very present in our daily life. And just to give you an example, because they are really famous because they produce a lot of secondary metabolites, then some of them, they are actually antibiotics. And I'm just gonna give you a few examples right now about some compounds produced by Streptomyces, and I'm sure that you all are familiar with these compounds. For example, one of them is streptomycin. This is easy, and I'm sure that it's produced by Streptomyces griseus, and I'm sure that you all use this in the lab to select your colonies. Another one that is actually more common that you take at home together with amoxicillin is clavulanic acid, and that's produced as well by another Streptomyces. And there are other compounds as well that they actually are colorful, and the mother organism in Streptomyces biology has been Streptomyces silicolor, and Streptomyces silicolor produces this compound that is called actinorodin that is blue, and as you can see, it can be accumulated on the surface of the colonies. Okay, so this is really interesting, but now I'm going to present my favorite mother organism, that is the one that we use at the lab, and this is Streptomyces venezuela. I think that you can guess where it was isolated. And Streptomyces venezuela looks like that, green, and it's famous because Streptomyces venezuela is the one producing chloramphenicol, an antibiotic that probably you use a lot in the lab as well. This is really interesting, but as Enrique said, I really like fundamental microbiology, and I think that the most fascinating thing in Streptomyces is that they are different from other bacteria, because they have an amazing life cycle, and this is what we study in the lab. I'm going to explain now the life cycle in detail because I'm going to go in some aspects of the life cycle. I'm going to go quite in depth. So Streptomyces bacteria, they sporulate, but they do it in a particular way that is completely different from Bacillus. And I'm gonna go through the life cycle starting first from a spore, okay? Then when the conditions are favorable, they germinate and they form a mycelium, vegetative mycelium. So mycelium is formed by these filaments that they are called hyphae, and these hyphae, they grow by tip extension. So there's no cell division in the vegetative state. So as you can see here in this picture, so you can see that sometimes some septa appear in the hyphae, and the function of this is just to make compartments in the hyphae to make viable the exchange of nutrients. But when the nutrients run out, they trigger sporulation, and the way that they do is they form these special structure, reproductive structures that they are called aerial hyphae, and these special hyphae, they grow up in the air. And at some point, they stop growing, and now the most fascinating thing is happens, that is like there's this massive cell division event, or synchronously. So they form these septa that they make regularly sized compartments, and in each compartment there's only one single copy of the chromosome. So they segregate the chromosome at this point, and eventually these septa they are different from the ones in vegetative growth, and these septa constrict and leads to cell-cell separation, and they form this chains of spores. So I want to point out that in the life cycle, and I'm going to go in detail of this later, in the life cycle there are two different cell divisions, two different types of cell division. As I said before, during the vegetative growth there's the crosswall formation, and crosswalls are the septa that I mentioned before, and this is just leads to compartmentalization of the hyphae. And during the sporulation, there's the sporulation septation. There is a proper cell division, like in other bacteria, and it leads to cell-cell separation and in the end, like spore formation. And one of the nice things of working with Streptomyces is that you can actually tell this life cycle just by looking at the colony. And in the case of Streptomyces Venezuela, the one that we use in the lab, so the colony looks like this, and you can tell that they produce aerial hyphae because the colony looks fuzzy and hairy, and you can tell as well that they are sporulating because all the spores in different Streptomyces, they have a pigment associated to the cell wall that in the case of Streptomyces Venezuela is this green pigment. This life cycle has been studied like for a few years now, and there are regulators that they have been described to control different transitions in the life cycle. So we divide these regulators into different classes, and I think that is a kind of a really nice way to call them, and now you will see why. So the first set of regulators actually control the development of the aerial hyphae, and these are called bolt regulators, because if you mutate these genes, Streptomyces is stuck in vegetative growth, so they don't produce the aerial hyphae. So the colony is not going to look hairy anymore, so that's why they are called bolt. And the second group of regulators are the white regulators, and these regulators control the cell division event during sporulation. And these regulators are called white because if you mutate these genes, they still produce aerial hyphae, but they are not able to form mature spores. So the colony looks white. I think that you can tell the difference between green and this colony white. Well, as I said, I've been working in a couple of projects in the lab, and one of the projects is focused on the aerial hyphae development, and the other one is focused on more in the cell division during sporulation. And I'm going to be talking about these two projects a little bit during my talk. And first, I want to talk about the first project that I'm working on is the aerial hyphae development. And so aerial hyphae is essential for sporulation in Streptomyces. And one of the things that is particularly so we switched to work with Streptomyces Venezuela instead of silicolor, because silicolor is not able to sporulate in liquid media, and Venezuela is able to carry out all this life cycle in liquid media as well. And I'm going to show a a movie later just to show you how we studied sporulation. And it helps when we want to actually address some aspects of the physiology with omics techniques. But now I'm going to be talking about like sporulation during solid media on solid media. So we can actually see in the electron microscopy Streptomyces growing the aerial growth. And as you can see here, this is a SCM picture taken by Kim Finley. She's the one in the bioimaging facility in the John Innes Center. She's an expert taking these beautiful pictures of Streptomyces, and she's the only one taking these beautiful pictures. And as you can see, we can see here that this is vegetative growth, and you see here the aerial hyphae growing, and some of them they have a spore already. But the mechanisms that the hyphae used to grow up in the air, I think is something really cool. And the hyphae can grow up in the air because they actually are surrounded by this sheath that is very hydrophobic, that is formed by two different families of proteins that are called chaplins and rodlins. And these proteins assemble on the cell wall, and they form the hydrophobic sheath, and then they form these very well organized structures. Sorry, the pointer is just this very well organized structure here, and that's why these proteins are considered amyloids. Okay, so this is easy to see by SEM pictures, and the hydrophobic sheath still remains in the spores. So the presence of this hydrophobic sheath makes Streptomyces colony really hydrophobic, and that's a feature that we can clearly see, and we can take these nice pictures as well. If you put some water just on the top of the colony, you can see these droplets staying there and not mixing with the colony. I'm gonna go in detail how this is regulated, but I'm gonna skip many details. But in general, there's a gene that is important for the expression of this hydrophobic sheath, and this is this gene. Is this gene here that is called Bolden. Okay, Bolden encodes an alternative sigma factor, and during vegetative growth, the expression of Bolden is repressed by another transcription factor that it doesn't matter right now. But when they are going to sporulate, Bolden expression is the repression is released. Bolden is expressed, and Bolden activates the expression of chaplins and rodlins and other genes involved in sporulation. And these proteins are expressed, exported to the extracytoplasmic part, and they assemble on the cell wall. And this is how the vegetative growth is controlled. This is the simplest model, let's say. As said before, the Streptomyces Venezuela can sporulate as well in liquid media, and that allows us to study these organisms by time-lapse microscopy. And I'm going to show here now a video, a movie just showing actually, even if it's a mystery for us why Streptomyces is going to express this hydrophobic sheath in liquid media, because it can't escape from the hydrophilic environment, but they do, and it seems that actually the cascade for sporulation in liquid media is exactly the same as in solid media, on solid media. So you will see here the spore. Okay, you will see there the spore. I don't know if you can see it in here. And this strain is the wild-type strain, and it's expressing the YFP from the promoter of one of these chaplins, okay? And you will see now that the spore is germinating, growing, creating this vegetative mycelium, and eventually you will see that they start to express this promoter, this chaplin. And if you follow, for example, this hypha here, so you will see that after expressing the hydrophobic sheath, they switch off the expression and they sporulate. It's still a little bit of a mystery for us why this is happening, but there are other studies actually suggesting the chaplains and rodlins are present during liquid media growth. This is a video again. So now you can. There are a couple of hyphae sporulating. I can't point in the video. I don't know why. Okay, so this is a very simple model how the hydrophobic sheath is regulated, but it's not that simple because the sigma factor, of course, is regulated by an anti-sigma factor. Probably, I mean, I'm not sure if plant people are familiar with this, but it usually, the activity of the sigma factor, sometimes alternative sigma factor, is controlled by anti-sigma factors. And in the case of Bolden, there's an anti-sigma factor that is just downstream the sigma factor gene, and it's called RSPN for regulator of sigma Bolden. So RSPN is a protein that inhibits the sigma factor. Okay. So my project focused first in the study of this anti-sigma factor and how this anti-sigma factor is controlling the activity of the sigma factor. And the first thing I did is just to mutate the gene of the anti-sigma factor. And in that case, we could see this phenotype, and this is something that is well described in our lab. So as you can see here in SEM pictures, you can see the wild type, this beautiful change of spores, and in the case of the anti-sigma factor mutant, as you can see, everything is spores. So RSBN is a breakthrough sporulation because it's inhibiting the activity of the sigma factor, and when you remove that break, the sigma factor is going to be free all the time, promoting sporulation. And this phenotype can be rescued by expressing the anti-sigma factor in trans. And I was able to see in liquid media as well this phenotype, and I'm going to show you how we studied this phenotype in liquid media. So what I'm doing here is just to label all the septa with the cell division protein FTSZ, that is a homologue of the tubulin. And as you can see, this is just to make you easy to understand what you're going to see in the time-lapse video. So during vegetative growth, you will see septa appearing in yellow, separated from each other, but it will be really easy for you to distinguish the hyphae that they are sporulating, because they form these beautiful FTSZ ladders. So this is the wild type, and in the wild type strain, you have to get used to these videos because I'm gonna show later, and but it's easy to see them. So you will see they are growing. You see the septa appearing, the vegetative septa, and now you will see that some of the hyphae, they are going to create these ladders that leads to sporulation. In liquid media, not all the culture sporulates. That's something that we don't really understand, but there's a control of sporulation only to certain hyphae. And in the case of the anti-sigma factor strain, where the anti-sigma factor has been deleted, as you can see here, this is a video, it's similar, so there's vegetative growth as in the other strain, the wild type strain, but suddenly, when they have to sporulate, all the hyphae is sporulating. Okay, indicating that in liquid media there's hypersporulation phenotype as well. And to prove that this hypersporulation phenotype is actually due to the overactivity of Bolden, the sigma factor, I introduced in this strain the promoter of this chaplain promoter controlling the YFP. As you can see in the wild type, this is the same time point. In the wild type, there's only a few hyphae expressing this promoter. In the Bolden mutant, there's no expression of the promoter, indicating the expression of this promoter is depending on this sigma factor. And in the case of the anti-sigma factor mutant, most of the hyphae actually are expressing the promoter. And if you can see, some is full of spores already. that was really interesting, and we wanted to know how the anti-sigma factor was inhibiting the function of the sigma factor. And for that, so it's easy to see that the anti-sigma factor is a big protein and is a membrane protein. So one of the things that is doing the anti-sigma factor is to recruit the sigma factor to the membrane, and in this way it prevents the sigma factor from binding to the DNA. In this case, we collaborate. Our collaborator Maria Schumacher in Duke University, North Carolina, in the U.S. She was able to crystallize the complex formed by the sigma factor and the anti-sigma factor domain of RSBN. And this is the structure of the protein. So we published this last year. And as bolded in blue, you can see the sigma factor, and in magenta, you can see the anti-sigma factor. Bolded belongs to these extracytoplasmic function sigma factors. And these sigma factors are like quite small. They only have 2 domains, and it's sigma 2 and sigma 4. And as you can see here, this is something that we were expecting. And in magenta, you can see the anti-sigma factor, and the anti-sigma factor was formed by 3 different helices. But one of the interesting things in this anti-sigma factor is that only the helix 3 was necessary to inhibit the sigma factor. So only the presence of the other 2 helices are just stabilizing the complex. So the presence only the 1 helix is enough to inhibit the function of the sigma factor. So Maria was quite excited about this structure because, I'm not a structural biologist, but she's one of the best, I think. So far, all the anti-sigma factor had been classified in 2 classes because all of them, they had quite similar structures. So the class 1 was formed by these 3 different helices stacked together with another helix, and the class 2, they were only formed by 2 different helices, quite small. But in the case of RSBN, actually it didn't look like the other ones. So this is, you can compare. So we were happy to see the RSBN define a new structural class of anti-sigma factor. And as I said, this anti-sigma factor the anti-sigma factor domain of the whole protein can inhibit the function of the sigma factor. But so then it was really interesting in my project as well to see but the protein is really big. So what is the function of the other part of the protein? Because that's only a small domain of the protein. And in this case, this is so this topology of the anti-sigma factor and the recruitment to the sigma factor to the membrane, it looks like other systems. For example, the one described in Bacillus, Escherichia coli, Mycobacterium tuberculosis. All these anti-sigma factors, what they do is they actually sense stress in the cell wall. And some of these systems are quite complex because they have other proteins that they are involved in recognizing signals and bringing other proteases together and everything. But there's something common in all these systems, and is that all of these anti-sigma factors are degraded by a common mechanism that is called regulated intramembrane proteolysis. So we thought that probably RSBN was subjected to regulated intramembrane proteolysis, and this is happening. This is a process that is carried out by 2 different proteases, and the process is that after sensing one signal, so there's one protease that is called site1 protease, and these are general proteases in the cell that they cleave the anti-sigma factor in the extracytoplasmic part. And then after this first cleavage, a second cleavage can happen that is within the transmembrane segment, and this one is carried out by this site2 protease. And this releases a cytoplasmic complex that is formed by the anti-sigma domain of the anti-sigma factor and the sigma factor. And in this case, the anti-sigma domain is very unstable and is degraded by general proteases in the cell. So we thought that was probably the case of RSPN. So the easy way to prove this is just taking samples along the life cycle of Streptomyces using an antibody anti-RSPN and see how it looks, the pattern of bands. So what I did is, so we have this super nice machine that is automated Western blot, and this is how it looks like. So I was happy to see that I could distinguish 3 different bands along the life cycle of Streptomyces, 3 main different bands. I'm not gonna go in detail because this is when the project got complicated, okay? But I was able to prove that these 2 bands correspond to the full-length protein, but the most interesting thing is that I see a processed form of the anti-sigma factor. Okay, so the next step is, it was like, okay, now we know that it's being processed, so let's have a look at the proteases that might be involved in this process. So Streptomyces, or actinobacteria in general, they have big genomes. So I tried to identify proteases that maybe that might be candidates to cleave the anti-sigma factor. The problem with the site 1 protease is the one that carries out the first cleavage is that in general they are conserved, and some of them they are signal peptidases, and so it was difficult for me to find like good candidates. Anyway, I mutated some of them. I didn't see anything. But the good thing is that site 2 proteases are very well conserved, actually even in eukaryotes. And in general, most bacteria, they have only 1 site 2 protease, but Streptomyces is cooler, so it's got 3. So I identified these proteases and that I called RIP1, RIP2, and RIP3 because in Mycobacterium tuberculosis they were named like that. And I made the mutants of these proteases. If one of these proteases was involved in degrading the anti-sigma factor, so and it's not present, I would see delayed sporulation because the anti-sigma factor would be for longer inhibiting the function of the sigma factor. So these are plates of with these mutants. This is a wild 3 different days, the same plate, and I put as a control the overexpression of the anti-sigma factor. And as you can see, the overexpression of the anti-sigma factor leads to delayed sporulation because in the day 2 the wild type is already white and the overexpression of the anti-sigma factor is still bold. And as you can see, it was really clear that the mutants of RIP2 and RIP3, they actually look like the wild type, but the mutant for the RIP1 was like the overexpression of the anti-sigma factor. So that was really cool. I was really happy to see the RIP1. This might be involved in the processing of the RSPN, the anti-sigma factor. So what I did as well is just to check the processing of these the processing of the anti-sigma factor in these mutants, and by Western blot again. But I mean, it's just forget about all these bands, just focus on this. And as you can see in the RIP1 mutant, there's so the processing band is missing. This is very sensitive, and right now I'm creating like double mutants just to actually see if some of these proteases actually might be redundant. So this is what I wanted to tell you a little bit from for the project that I'm doing regarding the aerial hyphae development. And now I'm going to talk about sporulation. Well, and in this case, I want to mention first that this is a project that I'm doing with Susan Schimpler, that she was a postdoc in my lab, and now she has her own group, and she's taking this research like her own, and it's really nice to work with her. And with Matt, that he's a postdoc in her lab. Well, and this is the 3 of us discussing the project over a pint in my favorite bar in Norwich. And I want to, this is not winter, as you can see. But first, I'm going to talk about sporulation, but I need to remind you how cell division happens in bacteria, and I'm gonna do it like in a very easy way, really simplified way. And well, cell division in all bacteria, in most of bacteria, depends on the cell division protein FDSZ. And FDSZ forms a ring in the middle of the cell, and this ring recruits all the proteins that actually lead to to constriction and cell-cell separation. But as I said before, and I've been like pointing this out quite a lot, in Streptomyces, cell division is special because we have two different types of cell division. So we have the proper cell division during sporulation, and we had this crosswalk formation during vegetative growth that they don't lead to cell-cell separation. And in this case, so this is just another scheme, just showing that during vegetative growth we had crosswalls formation, and these compartments contains several copies of the chromosome, and during sporulation we had small compartments just containing one copy of the chromosome. Something that happens during sporulation is that, okay, when they are going to sporulate, they are going to form thousands of septa. So one thing that must happen during this time is that all the cell division genes have to be overexpressed, and this is something that Matt did a few years ago, and he identified two transcription factors that they are involved in controlling the process of sporulation. These transcription factors are called white-A and white-B, and you know more or less the phenotype. If you mutate these genes, they are white. And these transcription factors, they control genes involved in stopping aerial growth, because the aerial hyphae has to stop growing before sporulation. Other genes that they are involved in late stages of sporulation, controlling expression of other genes involved in cell wall remodeling and everything. Of course, they control cell division genes. Actually, they directly control the expression of FDSZ. So what Matt did was a ChIP-seq analysis of these genes, of these transcription factors, and of course he found many genes that they weren't characterized. And one of them was interesting for us. Don't ask me why, but this one is, and it's the one that we call CEPH. Well, CEPH, the name is kind of a little bit of a spoiler, but I'm gonna go in detail how we found the function of this gene. And this is just a ChIP-seq showing showing the white-A and white-B binds specifically to the promoter of this gene. The first thing to do to see the function of a gene is just deleting the gene and just having a look if it has a phenotype or not. So Matt did that mutant, and it was surprising for us because that mutant actually could sporulate, but the colony looked like really weird. It wasn't green; it was more gray, and we thought that probably it was impaired in sporulation. But actually, the mutant could sporulate. But as you can see here in this SEM picture, the spores had like really big size, and they were very weird. And if you express this gene in trans, you can actually complement and restore the wild-type phenotype. So that was really interesting, and saying, okay, so we have really big spores, we don't have these small compartments anymore, so is it actually the dynamic of the formation of these ladders, FDSZ ladder, is it actually impaired in this mutant? And to do that, it was easy, just let's express FDSZ fused to the YFP and have a look in the time-lapse microscopy. This is a wild type. You've seen these movies before, but here is more in detail about two hyphae that they are sporulating. You see the formations of the FDSZ ladders that they lead to cell-cell separation and sporulation, formation of spores. So what is happening in the CEPH mutant? I mean, this is a little bit more complicated because you have more hyphae here. But as you can see, if you look in the here it's quite difficult to see, but if you look to the bright field, you can clearly see that some of the spores are not right. And if I stop this and I point this, so you can see here that in some of these, you can clearly see that some of the set rings, they form, but they just fade away. And that leads to spores that actually they are branched or simply just two spores fused. Okay, so it seems the CPH actually is doing something to FTSZ because the rings don't look stable anymore in this mutant, and so that's why the next step is to check, okay, where is CPH actually? So we fused CPH to the YFP, FTSZ to the M cherry, and as you can see, both of them they co-localize in the intercellular, in the sporulation septa and in the vegetative septa. And in this case, it was really easy for us to predict that probably these proteins actually directly interact. But as I said before, the Z rings recruits many other proteins involved in different processes during cell division, so probably it wasn't actually a direct interaction. Probably there was something else. So that's why we had a look at the protein, at the domain of organization of the protein, and I have to say that this protein is really conserved in actinobacteria, and actually it's been shown that this gene by transposon mutagenesis is essential in Mycobacterium. But the conserved part of the protein is in the N-terminal part, and this N-terminal part has a helix-on-helix motif. As you might know, the helix-on-helix motif is involved in DNA interaction, and the C-terminal part is unstructured, and it's got this predicted transmembrane helix. I'm not gonna show all the things I did about this, but I was able to prove that this protein is not binding DNA, because this is it will be like slides of negative results. But this protein doesn't seem to bind DNA, and so we were actually quite interested that probably this helix-on-helix motif is involved in protein-protein interaction, even if that's not the classical thought that you had for the helix-on-helix motif. And then I was able to prove as well that membrane is not an integral membrane protein, although it's localized in the membrane. But it seems that this localization in the membrane depends on the interaction with the membrane or with other proteins. Okay, so the easy is, so I did this GIS2 hybrid to check if actually this protein was interacting directly with FTSZ. And this is just GIS2 hybrid. Probably you are used to this, but if you see growth in this plate, it means interaction. Two interesting things we could see here is the CPH interacts with itself, and CPH interacts directly with FTSZ. I did truncations of the protein, and the interaction only was lost when I removed the N-terminal part where the helix-on-helix motif is. And okay, so now we knew that these proteins were directly interacting, and the N-terminal part was important for this interaction. It's actually, as I said, FTSZ formed this ring, and this ring depends on polymerization of FTSZ. Is CPH some way having some influence in the polymerization of FTSZ? And a way to study this is just by light scattering, by dynamic light scattering. So you will see these graphs, and what's happening here is that when FTSZ is in monomers, so you won't see light scattering, so it doesn't scatter the light that much. But when you add GTP, FTSZ is gonna polymerize, it's gonna hydrolyze the GTP, and it's gonna depolymerize at the same time. So that's why you see an increase in the light scattering, and it means the FTSZ is forming filaments and bigger particles, and they are scattering the light more. So this is how FTSZ on its own looks like. So I did this in the presence of CPH. I haven't included the negative control for CPH, but CPH doesn't scatter the light in the presence of GTP, so it doesn't polymerize. And in the case of the presence of CPH, you see that there's an increase in the light scattering. What makes us think that probably CPH is promoting polymerization in this case. I did as well, I purified the truncated versions of the protein, and I was happy to see that if I was using only the N-terminal part of CPH, the scattering looked very similar to the full-length protein, and if I was using the C-terminal part of CPH, the light scattering looked exactly like the FTZ on its own. But this increase in the light scattering doesn't tell us if it's actually just because they are forming big polymers, or it's just because there are more particles and they scatter more the light. So that's why, like, seeing is believing. So we went to transmission electron microscopy and negative staining, and we looked at how the filaments look like in the presence of CPH. This is, I'm not sure if you can see well, but this is FTSZ, and in the presence of GTP, you see these filaments. But in the presence of GTP plus CPH, there are more filaments, and they seem, I mean, we need to study this a little bit more in detail, but it seems that these filaments are actually more structured than FTSZ on its own. And then terminal part, we could see as well bigger filaments. And if we were adding just only the C-terminal part of the protein, it was like FTSZ on its own. So it seems actually the N-terminal part of the page is the one doing this effect in FTSZ polymerization. And I'm going to stop here, and I'm gonna tell you take-home messages from these two projects, and I've told you regarding the areal growth. I've told you that RSBN describes a new class of anti-sigma factors, and this anti-sigma factor is processed during development, and there's this conserved RIB1 protease that is involved in the processing of RSBN. And regarding the sporulation septation, we have described CPH. There is a novel cell division protein that is very conserved in actinobacteria. This protein interacts directly with FTSZ, promoting FTSZ polymerization. And this stabilization of the filaments is essential for the set reinformation during sporulation. And with that, probably some of you recognize that city. I want to thank Mark Partner, my supervisor, because he's very supportive, just giving me the freedom to do anything I like. Susan and Matt, because they are working with me in this CPH project that is going very well, and it's really nice to work with them, really nice people. Kim Finley, because she takes these beautiful SEM pictures of Streptomyces. Jake Richardson, that he helped me with the TEM of the FTS filaments. Maria Schumacher, our collaborator in the US, that she's the one who made the structure of RSBN, anti-sigma domain, and now she's working in the structure of CPH together with FTSZ. And with all that, well, the fundings, the BBSRC and Jonina Center, and with that, I'm happy to take questions. Thank you for your attention.
¿Preguntas para Félix?
Me ha encantado el formato de la presentación, muy bonita. Yo te quería preguntar cómo se induce la esporulación. O sea, tú dices, pongo la espora aquí y ahora le hago un vídeo.
No, nadie lo sabe.
¿No? ¿En absoluto?
No. De hecho, para los timelapse, nosotros lo que usamos es spent media. Los esporulamos en medio líquido, filtramos, cogemos las esporas y usamos el medio filtrado porque eso induce la esporulación antes.
O sea, que
hay señales.
Componente misterioso.
Sí, pero los medios son súper complejos y es imposible saber qué es lo que... Cuando le pones glucosa no les gusta y no esporulan.
Que, por ejemplo, para cualquier cosa que implique ómicas, o tú necesitas un timing, necesitas...
Pero es muy... sorprendentemente es muy... En torno a las 18 horas o así es onset of sporulation, están empezando a esporular. Y en líquido normalmente lo miras al microscopio y están fragmentados. Es la forma de darte cuenta de que están esporulando. Pero nadie sabe realmente qué es lo que desencadena, porque de hecho cuando tú usas el medio después de haber esporulado, ellos todavía, si lo usas, el spent media, ellos crecen vegetativos, entonces quiere decir que nutrientes tienen. No lo
Y
sé.
luego otra cosa sobre, bueno, el resultado negativo de que la helix turn helix no se une al ADN. En fin, a lo mejor no ha encontrado...
Hemos hecho ChIP-seq, hemos hecho EMSA,
Sí, pero que igual no has encontrado el componente misterioso que cambia conformacionalmente o lo que sea.
Puede ser. A ver, tenemos bueno, hemos hecho también colocalización con una proteína de estas asociadas con el ADN, HAP-S, que es
Ajá.
una histone-like protein.
Y nunca está allí.
¿Eh?
Y nunca está.
No, nunca colocalizan, pero, a ver, realmente nosotros vemos, no sabemos cuánta proteína está difusa, la vemos en el septo, pero no sabemos si la que está... Es muy difícil probar resultados negativos. Estamos haciendo todo lo posible, pero... Mucho. El N-terminal pad. Es, sí. A ver, no lo he puesto, pero hemos complementado el mutante con la proteína de Mycobacterium y complementa... es como el wild type. Así que probablemente tenga incluso la misma función en Mycobacterium. En Corynebacterias es un poco más diferente en el C-terminal, pero...
A ver, Félix, bueno, a mí me ha encantado el seminario, como todas estas cosas de microbiología diversa. Aquí en este sitio, una cosa fundamental parece que es evitar los núcleos cuando hace el septo.
Sí.
Un mecanismo de ese de oclusión. ¿Qué mecanismo?
No se sabe. No tienen el MIN y, bueno, tienen PAR-B y eso. Y es que no está muy bien, no se sabe realmente. Se han propuesto dos proteínas, SGB, SGA, que son las que reclutan el anillo, que posicionan el anillo. Eso, pero en el laboratorio ahora Susan está trabajando con eso y parece que no hacen eso. No se sabe realmente cuál es el mecanismo porque no tienen este mecanismo de exclusión. No se sabe. Nosotros al principio, cuando vimos que podía unir ADN, dijimos, probablemente sea esta la proteína clave pero realmente no parece que sea así. Podría ser, pero no sabría decirte. Realmente creemos que la proteína puede tener algún tipo de post-translational modification. Perdón, pero es que suena como un poco repipi, ¿no? En plan pero creemos que puede ser porque si te fijas un poco es esencial para formar para la estabilidad de los set rings durante la esporulación, pero también las proteínas localizan en las crosswalls en el crecimiento vegetativo, pero no sabemos realmente y ahí, digamos que no hay constricción de los anillos. Tenemos algunas evidencias de que la proteína probablemente tiene algún tipo de modificación durante vegetativo y esporulación. Entonces podría estar involucrada en algo relacionado con la segregación de cromosomas, pero no tenemos ninguna evidencia para eso, pero sí para las modificaciones posttraduccionales. Puede ser, sí, sí, sí. Es que cuando hacemos Western blot vemos que baja. La banda baja, pero yo intento hacer cosas de fosforilación, pero no he visto nada.
Bueno, yo me sumo a las felicitaciones, me ha parecido un seminario magnífico. Me han encantado las fotos de microscopía y por eso te doy la enhorabuena. Sí, 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 sí.
Esas están en mi carpeta de Eye Candy Images.
A mí me ha llamado mucho la atención otro aspecto que a lo mejor no sé si se habría incidido, es en la culminación de la sextación. ¿Por qué las esporas culminan la sextación? Sin embargo, cuando está el crecimiento vegetativo de la hifa... ¿Habéis estudiado si...
No, nadie sabe eso.
Los genes tardíos de la división celular, ¿cómo están regulados esos genes? Porque se deben de estar regulados seguramente por estos factores que regulan...
White A y White B regulan casi todo. FTCW, FTCI, un montón. Pero claro, realmente no sé decirte porque los regula, o sea, activa la transcripción de esto, pero realmente porque necesitas más proteína en ese momento, porque vas a hacer muchísimo septo, pero no se sabe realmente cuál es la diferencia entre un septo, el crossword y el sporulation septa. No.
Porque durante el crecimiento vegetativo sí que se ve expresión localizada de FTCZ, que es como el primer gen, la primera proteína que interviene en el posicionamiento del septo, ¿no? Pero debe de haber algún punto a partir del cual ya no se expresan todos los genes de la división tardía, ¿no? En la IFAS vegetativa.
Es que el problema viene siempre. Es muy difícil responder esas preguntas porque cuando tú estás creciendo un cultivo, ¿vale? Eso siempre es una mezcla, siempre. Y ese es el problema que estoy teniendo también con el otro proyecto del procesamiento. Yo veo bandas, ¿vale? Pero probablemente unas bandas están solamente en estadio vegetativo y otras están en el esporulando, pero es que no hay forma de separar no hay forma de separar las hifas unas de otras y lo he intentado filtrando, pero es que no siempre sale lo mismo. Entonces, y ahora se han descrito algunas proteínas que están, solamente hay una proteína que se encuentra, que es específica de los septos de la esporulación, y es una dinamina que promueve la dinámica del set rings y eso. A ver, tenemos pull down con esa proteína, pero hay muchas proteínas ahí, o sea, es difícil.
Que hay que apretar el botón.
La audiencia afuera está diciendo, ¿qué están preguntando?
Félix, sobre todo, primero felicitarte. Yo como amante de la regulación génica asociada al desarrollo me ha encantado el trabajo y estoy muy lejos de la microbiología.
Pero es desarrollo.
Pero es desarrollo. Y me ha hecho recordar el dominio HTH, de que no tengo ninguna idea, pero viendo lo que has puesto de unión ADN y luego interacción de proteínas, y los fenotipos que estás buscando una explicación, un mecanismo, me ha hecho recordar que uno de los factores de transcripción con los que trabajamos son de la familia HLH. También son dos hélices, pero en plantas los VHLH, la parte que une ADN importante para la unión al ADN son los aminoácidos básicos que hay delante. Y la HLH lo que son dominios de dimerización. Entonces, en plantas existen algunos HLH raros que no unen el ADN, lo que hacen es secuestrar a los factores de transcripción para, digamos, titularlos y regular negativamente su unión al ADN. Entonces, podría ser una explicación, ¿no?, que vuestra proteína estuviera interaccionando con una que sí una al ADN y que sea un mecanismo negativo de represión de la expresión génica.
Sí, puede ser, es verdad. Creo que lo tienes que pulsar todo el rato. ¿O?
La función que sí que he demostrado claramente es que ayuda a FTZ a formarse, ¿no?
A formar.
Cuando estás solo, de hecho, parece ser que se polimeriza de una manera y cuando lo tienes con tu proteína como más lineal y más recto. ¿Tenéis imágenes al microscopio electrónico o algo así que os muestre y en qué consiste realmente? Eso sí sería muy interesante, ¿no? Porque las proteínas que ponéis son puras.
Sí, sí.
Sin modificar.
Sí, además no tienen ningún tag ni nada. Yo les quito el tag. Claro, lo que pasa que eso es difícil porque esto es in vitro realmente. Luego la polimerización de FTSZ in vivo tiene otros componentes también que regulan. Entonces es un poco difícil. Es difícil sacar conclusiones de sí, promueve polimerización, pero no sabemos realmente. A ver, María Schumacher está trabajando en la estructura y quiere hacer cryo-EM con los filamentos en presencia de CEPH. Probablemente eso nos dé algún tipo de por ahora tiene cristales de CEPH solo. Cristales con FTSZ yo no tengo mucha esperanza porque polimeriza y no cristaliza bien. Entonces, pero ella lo está intentando y ella todo lo que se propone lo consigue, créeme.
Porque sería interesante ver cuál es el mecanismo.
Sí, claro.
¿Le ayuda a romper el GTP o es que se une a ella?
Claro. Hay algo que no he mostrado aquí, es que normalmente vemos que aumenta la actividad GTPasa de FTSZ. Pero creemos que realmente eso es algo indirecto porque tenemos más filamentos. Porque una de las cosas claras es que si hay muchísimos más filamentos, poniendo la misma concentración de... con FTSZ tuvimos problemas al principio para ver los filamentos, me costó mucho optimizar eso, pero sin embargo siempre que estaba CPH es que eran fotos preciosas y era bueno, pero es que necesito el control. Y al final, lo conseguí simplemente incubando el tiempo que tenía que incubar y eso, pero sí.
¿Más preguntas?
Sí, yo tengo una pregunta general, muy general. Y es, ¿dónde viven estas bacterias y en qué condiciones evolucionan?
Pues Venezuela se aisló de Lima. No, de Lima no. De Caracas. No, pero yo no sé por qué digo yo Lima. Yo creo que leí alguna vez Lima. Digo, ¿por qué es Venezuela entonces? Bueno, el caso es que de la fruta. No, pero viven en el suelo, generalmente, sí. ¿Y en qué esporulan? El problema... Venezuela es el único que... Venezuela y Griseus son los únicos que esporulan en medio líquido. El resto tiene que ser siempre en sólido. Entonces, una de las cosas que pasaba con Silicolor, que era el organismo modelo, es que nunca se podía hacer time-lapse o algo porque tenías que coger todo de una placa. Nosotros siempre decimos que probablemente Venezuela viva en el agua, pero normalmente están en el suelo y son muy abundantes. Yo creo que de las más abundantes en el suelo. Pero no sé cuándo esporulan realmente. Si deben estar esporulando todo el rato porque ahí tendrán competencia.
No sé si lo he entendido bien, pero ¿el proceso se inicia con las proteasas? Cuando las proteasas cortan...
No, realmente el proceso de esporulación se inicia, ¿vale? Enseñé al principio dije que Bolden estaba reprimido por otro factor de transcripción. Pues ese factor de transcripción se llama BOLD y une cycled agent P, ¿vale? Y cuando está unido con cycled agent P, inhibe todo un montón de genes, cientos de genes. Y en un momento, que no se sabe cuándo, los niveles de cycled agent P disminuyen y eso hace que dejen de unir cycled agent P y se libere del ADN y se activen un montón de genes. Y uno de ellos es Bolden. Y Bolden sería el siguiente que activa, es como una cascada. Después va Bolden activa otro que se llama Bold-M activa los white, así.
Pero
Uy.
Bold-M es secuestrado en la membrana.
Sí,
Las proteasas hacen que se liberen.
sí.
Pero ¿influyen positivamente entonces las proteasas en el proceso de estructuración?
Sí. Sí, porque necesitas liberar el...
Y al estar en membrana, ¿podrían estar sensando estímulos externos?
Sí. Una de las cosas que era en mi proyecto era determinar cuál es la señal que... lo he intentado. Pero, o sea, he intentado... Es que he hecho muchas cosas en ese proyecto que no han ido a ninguna parte porque... Pero puede ser que la propia sheath, puede ser que eso sea lo que hace que se empiece a degradar el antisigma.
Ya has hecho bastante, ¿eh? Está muy bien.
En ese he hecho mucho. Bueno, quería...
La parte de la proteasa, son metaloproteasas.
Sí. Estas son es una clase de metaloproteasas que tienen el sitio activo en el primer hélice transmembrana y el sitio activo es HXE, es histidina, otro aminoácido glutámico.
No se tiene idea de lo que pueden, si sensan algo, aparte de ese metaloproteasa, porque lo normal es ese tipo de metaloproteasas normalmente tienen algo para un sensing.
Sí, la que...
Y por ahí estaría un poco la clave, ¿no?
Sí.
De todo lo que es
Esa, la que me dio el fenotipo, esa es la que, digamos, está conservada en todas las bacterias. Y esa tiene un dominio PDZ en la parte extracelular, que eso puede ser para interacción con otras proteínas y sensar cosas. Yo siempre creo que es que que lo que sensa la señal es otra proteína. Y luego las otras dos, RIP2, esa es... No veo ningún tipo de dominio, pero en la RIP3 hay otro dominio, pero es intracelular. Y el problema de esto es que estas proteasas probablemente degraden otros antisigma y Venezuela ECF tiene 27. Entonces, a ver, yo ya tengo los dobles mutantes porque hacer mutantes limpios en Streptomyces sin ningún tipo de cassette es muy difícil. Se tarda mucho tiempo. Y tengo los dobles mutantes y estos dobles mutantes están muy afectados en el desarrollo, pero realmente no sé si es por algo indirecto. Es que los he conseguido. La semana pasada conseguí los dobles mutantes.
Y hay una cuestión, Pablo, que me ha parecido interesante de lo que has dicho, aquellas que esporulan en medio sólido, de las que lo pueden hacer en líquido. Entonces, bueno, no sé, es algo que suele ocurrir, es decir, cuando tú tienes un medio sólido, la adquisición de los nutrientes está limitada espacialmente. Es decir, donde tú tienes esos nutrientes ahí se van agotando, mientras en un medio líquido, pero como hay una mezcla, eso es mucho más uniforme. Entonces me pregunto si puede ser simplemente que en estas de medio sólido, por un lado, se queden, habría que estudiarlo un poco más en detalle eso, pero claro, puede que la restricción nutricional empiece mucho más corto que cuando tienes medio líquido, que como aquello va mudándose al final en esas especies, pues eso no ocurre, o no sé si ocurrirá al final, al cabo de un tiempo mucho más. Mientras en estas que esporulan, pues, o pueden ser que simplemente tengan alguna capacidad de poder asimilar estos nutrientes de otra manera, o bien esto que hablaba un poco antes de que si eran más hidrofóbicas o hidrofílicas, si hay algo ahí que hace que al final los nutrientes tampoco cedan de alguna manera, ¿no?
No sé.
Y por lo que dice el GMP la restricción más fuerte probablemente sea el carbono, ¿no?
Sí, a ver, yo creo que tiene que ver algo con la fuente del carbono. Porque depende... Sí, porque si los creces en medio mínimo, que puedes controlar qué es lo que le pones, por ejemplo, no esporulan si le pones glucosa, bueno, la glucosa es bastante rica, pues a lo mejor, pero no esporulan, o sea, semanas. Venezuela y te esporulan 3 días. Y luego si le pones maltosas, sí que... La maltosa les encanta. Ese es su azúcar. Bueno, les encanta. A nosotros nos encanta que les guste porque esporulan, pero a lo mejor no les gusta tanto a ellos. Pero pero es pero es que yo cuando trabajaba con Ana Faena, pues obviamente es poliploide, pero es que estos tienen unos genomas y, a ver, para ser bacterias son 9 megabases. Si te paras a mirar cosas de transportador, es una locura. Son bastante complejos. A mí me gusta hablar. Vale, gracias.
Muchísimas gracias, Félix, y
Gracias.
que te siga yendo tan bien.
Gracias. Bueno, pues no he contado casi nada, porque la primera parte siempre me trae amargado, porque no se ha hecho tantas cosas.